Hello children. Today we will talk about the second step in aerobic respiration, the Krebs cycle. In this cycle, we will also learn about the amphibolic nature and the anaplerotic reactions associated with it. Krebs cycle has a total of 8 steps which include various types of reactions such as condensation, isomerization, decarboxylation, and hydration. Looking at the steps one by one, step 1 is the condensation type where a 2-carbon acetyl coenzyme A is converted into citrate. In the second step, isomerization of citrate forms isocitrate. The following reaction is oxidative decarboxylation of isocitrate to oxoglutarate, also known as alpha ketoglutarate. This reaction is extremely important as it involves not only a multi-enzyme complex, but there is also reduction of NAD to NADH with the loss of carbon dioxide. Step 4 is oxidative decarboxylation of alpha ketoglutarate to succinyl coenzyme A. This step is similar to the previous one in terms of the involvement of multi-enzyme complex and production of carbon dioxide and NADH. Step 5 is conversion of succinyl coenzyme A to succinate. This results in production of one molecule of ATP and coenzyme A. Please remember, this ATP production does not involve any electron transport chain and therefore is substrate level phosphorylation. Step 6 is succinate being converted to fumarate with the help of a multi-enzyme complex and FADH is produced. Here I would like to add that the enzyme involved is succinic dehydrogenase. This enzyme is unique to the Krebs cycle because this is the only enzyme which is found in the inner mitochondrial membrane. All the other enzymes of Krebs cycle are present in the matrix. Next step is fumarate being converted to malate. This is a hydration reaction. The last reaction, where malate is oxidized to OAA, is again catalyzed by a multi-enzyme complex and NADH is formed. So, these were the 8 steps which form the Krebs cycle. Now, let us look at the energy status of the glucose molecule which has been processed through glycolysis and link reaction and is now also processed in Krebs cycle. In Krebs cycle, you have production of 3 NADH, 1 FADH, and there is production of 1 ATP. Mind you, as 2 molecules of pyruvate have been processed or pushed into Krebs cycle through link reaction, we will have to multiply all these by 2. And therefore, we have 6 NADH, 2 FADH2 and 2 molecules. In link reaction also, we have seen the formation of NADH and ATP. Similarly, here also we will multiply the net result by 2 because 2 molecules of pyruvate have been produced. On the other hand, in glycolysis, we have net production, as we said, of 2 ATP and 2 NADH. Next, we would be talking about the salient features of Krebs cycle. Let us look at the salient features of Krebs cycle. The acetyl group of acetyl coenzyme A, which is produced through link reaction, is completely oxidized through 8 steps of Krebs cycle. The energy is conserved at least in 3 steps as NADH, In one step as FADH2, there is also production of 1 ATP through the substrate level phosphorylation. Two molecules of carbon dioxide are released in the process. The cycle was put together by Hans Krebs after the reactions involved in oxidation of acetyl coenzyme A were discovered by various physiologists and is therefore commonly known as Krebs cycle. It is also known as citric acid cycle since the first product is citric acid or citrate. It is commonly also known as TCA cycle because the citric acid, first stable compound formed in the cycle, has 3 carboxyl groups. One complete round of cycle with 8 steps generates 2 carbon dioxide molecules, 3 NADH, 1 FADH2, and 1 ATP. Since 2 molecules of pyruvate are fed into the cycle, it gives us a total of 4 carbon dioxide, 6 NADH, 2 FADH2, and 2 ATP molecules. Krebs cycle is a hub of cellular activity. It is because the cycle generates energy-containing compounds such as NADH and FADH2. Also, because it feeds intermediates such as citrates and alpha-ketoglutarate into various metabolic pathways. Since Krebs cycle operates in the mitochondrial matrix, all products are formed inside the matrix. From this matrix, the compounds are either consumed or they are transported out of the mitochondria into the cytosol for other metabolic pathways. Krebs cycle is associated with amphibolic nature, and some reactions are also of the aneuplerotic nature. The net equation of the Krebs cycle is given in the slide. Before we wind up our discussion on Krebs cycle, let us look into some unique features of this cycle. The first is the molecule acetyl coenzyme A. Acetyl coenzyme A is an important molecule processed through this cycle and is said to be hub of metabolic activity. It is also referred to as a gateway molecule because it is connected to carbohydrate, protein, and fat biosynthetic pathways. As we all know, acetyl coenzyme A, a 2-carbon compound, is pushed into mitochondrial matrix for oxidation through Krebs cycle. Acetyl coenzyme A is produced from pyruvate by the activity of PDH through the link reaction. In Krebs cycle, this molecule is processed to generate energy in the form of ATP and energy carriers in the form of NADH and FADH2. Acetyl coenzyme A is also produced in plastids and is processed into fatty acids, seed oils, and cutins. It is a building block of long-chain fatty acids. Acetyl coenzyme A is also the building block of isoprenoid units, which are used for the biosynthetic pathways of hormones such as gibberellins, cytokinins, and abscisic acid. Isoprenoid units are also used for the production of carotenoids and secondary metabolites. Amino acids also, some of them require acetyl coenzyme A. As an example, we have oxaloacetic acid. This intermediate provides skeletons of carbon for the amino acid aspartate. Acetyl coenzyme A also is known to donate acetyl groups in protein acetylation. The next unique feature of Krebs cycle is its basic nature of anaplerotic reactions. Anaplerotic reactions are the fill-up reactions To simplify, let us look at the example of oxaloacetic acid. This compound is converted to citrate in Krebs cycle. But as we know, OAA is also used to form PEP, and the enzyme used is PEP carboxykinase. Now, if this oxaloacetic acid is taken away from Krebs cycle and is not replenished, the Krebs cycle would not operate. Therefore, we need to fill it up or regenerate OAA back. This is possible by converting PEP into OAA through PEP carboxylase. These reactions have been done under fate of PEP, and the link is provided in the description box. There are several other examples of anaplerotic reactions, and citing one more, there is the example of malate or malic acid. In Krebs cycle, malic acid is converted to oxaloacetic acid. But malate can also be converted to pyruvate by malic enzyme in the cytoplasm. This is a diversion, therefore, of the intermediate. If malic enzyme is more active, malate will not be formed and will not be readily available for the Krebs cycle, which means the Krebs cycle will come to an halt. Therefore, to replenish malate, we should have anaplerotic reactions which can refill malate. The first is where malate for Krebs cycle can be obtained from PEP. PEP can be directly converted to OAA by PEP carboxylase, bypassing pyruvate formation. This I have already mentioned. In the second reaction, pyruvate can be carboxylated to oxaloacetic acid, and oxaloacetic acid can then continue the Krebs cycle. The enzyme for this is pyruvate carboxylase. We normally know that PEP can continue to produce pyruvate through glycolysis, and in which case malate need not be diverted. So these were some of the reactions. Similarly, you have the example of alpha ketoglutarate. If this is taken away from Krebs cycle to form glutamate, then glutamate synthesis will deprive Krebs cycle of the intermediate alpha ketoglutarate. But in the fill-up reaction, glutamate can be deaminated to form alpha ketoglutarate. There are certain amino acids such as arginine which can be degraded to glutamate, which can then produce alpha ketoglutarate. So these are the fill-up reactions. We also know that Krebs cycle is known to have an amphibolic nature. Now, amphibolic nature means that it should be able to provide the build-up reactions or the anabolic reactions and the breakdown reactions or the catabolic reactions. The simple example is citrate. When it is pushed through 8 steps of Krebs cycle to generate oxaloacetic acid, it is a catabolic pathway. However, if citric acid is used to build up fatty acids, we are talking about some build-up reactions, and therefore the process becomes anabolic. Similarly, breakdown of alpha ketoglutarate to succinyl coenzyme A is catabolic, which occurs in Krebs cycle. But if you take away alpha ketoglutarate as an intermediate from the Krebs cycle to build up amino acids, it is an anabolic reaction. Similarly, citrate builds up fatty acids, and PEP is for synthesis of sugars through gluconeogenesis. Before we wind up our discussion, finally on Krebs cycle, we need to know the regulation of Krebs cycle. Krebs cycle, like glycolysis, is a highly regulated cycle, And we should know that it is the ratio of NAD to NADH and the ratio of ADP to ATP which will govern whether the cycle should continue to operate or the intermediates may be pushed into other pathways. Therefore, we can summarize by saying Krebs cycle sits at the intersection of converging and diverging pathways and is very unique in plant metabolism. Next, we will be talking about electron transport chain.